Our body is made up of 3 different types of muscles: smooth, cardiac, and skeletal. Today we will be discussing skeletal muscle, its different structural components, and how a muscle contracts. First, let's discuss the basis of what skeletal muscle is made up of. Skeletal muscle is made up of fascicles. Fascicles are bundles of myofiber, also known as muscle fiber or muscle cell. One myofiber is made up of many myofibrils. Myofibrils are made up of myofilaments. A single myofilament is represented by the single green pipe.
A sarcomere is a contractile unit of a striated muscle composed of myofilaments. Myofilaments are composed of thick and thin filaments. Thin filaments are represented by the black and white red dots. With red dots, the red dots represent troponin. The black represent the tropomyosin, which covers up binding sites for the myosin head. And the pearls represent the actin molecules. Thick filaments are represented by the brown sticks with the leafy projections. The projections on the thick filaments are myosin heads, which form the cross bridge that interact with the thin filaments. A sarcomere goes from Z disc to Z disc. In the middle, we have the M line, or H band, that connects all the thick filaments. Notice, only thick filaments are present. No thin filaments are found here. The A band is composed of both thick and thin filaments, as well as the H band. The I band extends past the Z band on either side. Notice there are only thin filaments in the I band. Sarcomeres, which are made up of myofilaments, combine to make myofibrils, which make up muscle fibers. Muscle fibers are protected by a plasma membrane called the sarcolemma.
Sarcolemma, represented by the pink, surrounds the muscle fiber. The muscle fiber is made up of many myofibrils, which is represented by the red. In between the myofibrils are mitochondria, which are represented by the green pipe cleaners. Surrounding the red is the sarcoplasmic reticulum, which is shown by the blue tube-like structure that runs longitudinal on the myofibrils. The function of the sarcoplasmic reticulum is to store calcium and release it on demand when the muscle fiber is stimulated to contract. The T-tubule, which is represented in white, are the elongated tubes which are an extension of the sarcolemma. T-tubules are found deep in the muscle cell and allow for the cell to become more positive on the inside than it is on the outside, which is known as depolarization. Once the muscle is ready to contract, the calcium stored in the sarcoplasmic reticulum is released, causing the sliding of myofilaments. Before the process of muscle contraction can begin, a change in electrical potential must take place. This is known as action potential. Once action potential travels down the axon, the calcium channels are opened, allowing calcium to flow in. Calcium enters the axon and travels down, pushing the vesicles filled with acetylcholine into the cleft. The acetylcholine attaches to receptors on the motor end plate, opening them to allow sodium ions to enter and join the potassium ions already there. After the receptors open, acetylcholinesterase breaks down the acetylcholine, and the choline is taken up by the vesicles to be turned into acetylcholine again. Once the sodium ions enter the muscle fiber, depolarization occurs, and calcium ions, which were held by calsequestrin, are released from the sarcoplasmic reticulum and attached to troponin, pulling the tropomyosin off the active sites on the actin.
Myofilaments are made up of thin and thick filaments. The thin filaments are made of actin, and thick filaments made up of myosin. Actin is known as a contractile protein. A contractile protein is one that generates force during a contraction. As shown by the red and orange model, actin takes on the structure of a double helix that looks similar to 2 intertwined pearl necklaces. Actin also makes up thin filament. As you can see, the blue dots on the actin molecules represent a myosin binding site where a myosin head may attach. Myosin function as a motor protein. Motor proteins push or pull cellular structures to achieve movement by converting chemical energy to ATP. Myosin is represented by the purple and blue model, with the myosin heads starting out in yellow toward the M line on the sarcomere. Each myosin head has an ATP binding site shown on the model as black dots on the myosin heads. This is where an ATP molecule may attach. Both tropomyosin and troponin are known as regulatory proteins. These proteins assist in switching contraction on and off. Tropomyosin is represented by the green wrapped around actin. In relaxed muscle, the myosin head is blocked from attaching to the myosin binding site on actin due to tropomyosin covering the binding sites. Troponin molecules on the structure hold the tropomyosin in its place. This is demonstrated with the yellow circles underneath edges of the tropomyosin offering support. When calcium molecules bind to troponin, it changes the shape, therefore moving the tropomyosin away from the binding site. At this point, the myosin head attaches to the binding site on actin and a muscle contraction begins. ATP molecules stemming from the mitochondria are made up of the combination of molecules adenosine phosphate and phosphate plus energy, together creating one ATP. When an ATP molecule is attached to the myosin head is in a low energy configuration and attached from the thin filament. When energy from the ATP attaches to the myosin head, leaving an ADP, this puts the myosin head in a high energy configuration. At this point, the myosin leaps over and attaches to the binding site on actin and pulls the thin filament along with it as it swivels down to a low energy state. As another ATP comes along and attaches to the myosin head, myosin then detaches from the thin filament and the process repeats itself.